Welcome back everyone. This is the 9700 February-March 2021 paper 22. You guys wanted the paper 22 series 2, so here I am answering your call. If you are new, you know what to do. Like, comment and subscribe. And also, if this video does get enough likes, I will drop another paper 2 video soon. Let's get started. Question 1. Figure 1.1 is an image of a transverse section through the trachea of the gas exchange system. Write the names of structures X and Y on figure 1.1 in the boxes provided. They have given an image of a transverse section through the trachea, and over here you simply have to remember which parts represent which. So they have already labeled the lumen, the ciliated epithelium and a layer called perichondrium, and they are asking us to label X and Y. X is nothing but a mucus gland which produces mucus. Y is cartilage. And over here you simply have to memorize which tissues represent which. So again, X is mucus gland, Y represents cartilage. B. The perichondrium contains collagen fibers composed of collagen molecules. Collagen is an example of one type of biological molecule. State the name of the type of biological molecule of which collagen is an example. Collagen is a fibrous protein used for structural roles. So you could either say collagen is a protein or you could mention fibrous protein. Since they're asking the name of the type of biological molecule, we should state whether it is a protein, whether it is a carbohydrate, whether it is lipids. Since collagen is a fibrous protein, we can simply say fibrous protein or just protein. Part C. Some structures in the gas exchange system are listed in alphabetical order in table 1.1. Write yes in the box provided if the structure contains smooth muscle. Write no in the box provided if the structure does not contain smooth muscle. Out of all these given structures, only alveolus doesn't contain any smooth muscle. All the others does. Therefore, for alveolus, you should put no. For bronchiole, bronchus and trachea, it will be yes. Question 2. Starch molecules are the main storage molecules in many types of cereal grains such as the grain of the barley plant. Part A. When the seed inside a barley grain germinates, genes coding for digestive enzymes are switched on. The enzymes that are synthesized catalyze the hydrolysis of storage molecules such as proteins and starch. Explain what is meant by a gene. A gene is simply a length of DNA that codes for a polypeptide or protein. You could also mention the gene in terms of a sequence of nucleotides. So you could mention gene is a sequence of nucleotides that codes for a polypeptide or protein, or either you could mention gene is a length of DNA that codes for a polypeptide or protein. Since this question is 2 marks, 2 points are required, and the 2 points are length of DNA or sequence of nucleotides, and the other mark is for codes for polypeptide or codes for a protein. Part 2. The hydrolysis of proteins in the barley seed produces amino acids that can be used in the synthesis of the proteins required for formation of the seedling. Figure 2.1 is an incomplete diagram of the molecular structure of the smallest amino acid, glycine. Each molecule of glycine has 2 carbon atoms. Complete figure 2.1 to show the molecular structure of glycine. Key thing you should remember is that all amino acids, no matter what the name of the amino acid is, all amino acids have 3 constant regions: a hydrogen atom, a NH2 group and a COOH group or a carboxyl group. Therefore, in this incomplete diagram, NH2 is missing and COOH is missing. Therefore, in order to complete this incomplete diagram, you have to draw 2 key parts. One is the NH2 group, which is represented on the left side of my incomplete diagram, and the other is the COOH group. The carbon is already given, but you have to draw the double bond O and a single bond OH attached to the second carbon. Again, a key thing you should always remember is that do not forget to place a double bond when you are joining the oxygen atom towards the second carbon atom, that is carbon double bond O, and then a single bond towards the OH group. That is how you draw the COOH group. There should be carbon double bond oxygen and then single bond OH. And a hydrogen atom is already given. The fourth group is simply a R group. It can be a hydrogen atom or any other R group. Key thing to remember is that all amino acids have 3 constant regions. What are they? A hydrogen atom, a NH2 group and a carboxyl group or a COOH group. Part 3. Starch is a mixture of 2 different molecules. Name these 2 molecules. What you should know is that starch is made up of 2 different molecules which are known as amylose and amylopectin. Amylose only has 1,4 linkages, whereas amylopectin is a branched molecule. It has both 1,4 and 1,6 linkages. If a molecule has 1,6 linkages, that means it is branched. Both are made up of alpha glucose and both are polymers. And amylose and amylopectin molecules, they both form part of starch. Hence the two molecules are amylose and amylopectin molecules. Two of the enzymes synthesized by the barley seed are alpha amylase and maltase. These are involved in the hydrolysis of the stored starch during seedling formation. In the food industry, the starch extracted from barley seeds is used in the production of sugar syrups. Figure 2.2 summarizes the reactions catalyzed by alpha amylase in the production of maltose syrup and by maltase in the production of glucose syrup. Part B. Some of the substances shown in figure 2.2 are listed in table 2.1. Complete table 2.1 to identify which of the terms polysaccharide, monosaccharide and macromolecule apply to each of the substances listed. Use a tick if the term applies and a cross if the term doesn't apply. Put a tick or a cross in every box. So you have to follow the guidelines given. You have to put either a tick or a cross in each of the boxes given. And if you look at the table, the substances are glucose, maltase, maltose and starch. Glucose. Well, glucose is not a polysaccharide but a monosaccharide. Hence, there should be a cross under polysaccharide and a tick under monosaccharide. Glucose is also not a macromolecule because monosaccharides are the smallest sugar units and it cannot be a macromolecule. Therefore, glucose is not a macromolecule. Let's look at maltase. Maltase is an enzyme and all enzymes are made up of proteins and they have a tertiary structure. Therefore, maltase doesn't belong to polysaccharide or monosaccharide as it is not made up of sugar units but it is a protein. Hence, it should belong to macromolecule because enzymes are made up of proteins and since they have a tertiary structure, yes, they do form part of the macromolecule. Maltose. Maltose is not a polysaccharide and is not a monosaccharide. It is a disaccharide. Since disaccharide is not given, you have to put a cross under polysaccharide and monosaccharide, as well as maltose is a disaccharide and it cannot be a macromolecule. Macromolecules are those which are polysaccharides. Therefore, another cross under macromolecule. Starch. Starch is a polysaccharide and starch is a macromolecule because all polysaccharides are macromolecules. It is not a monosaccharide, hence there should be a cross under monosaccharide. When producing sugar syrups, there are advantages in using enzymes extracted from microorganisms. For example, some enzymes extracted from microorganisms are heat stable. Heat stable enzymes are used to increase productivity because the reactions can be carried out at higher temperatures. Part C. Suggest one other advantage of using enzymes obtained from microorganisms rather than enzymes extracted from barley seeds in the production of sugar syrups. They only asked to suggest one other advantage. However, I have listed some advantages which you could mention. You could choose either one of the advantages given and note it down. So the possible advantages are: use of microorganisms allows large scale production of enzymes because, well, in microorganisms they can carry out multiple divisions very rapidly, hence allowing large scale production. You could also mention that it is easier to extract from microorganisms rather than extracting from barley seeds. Another possibility is that using enzymes obtained from microorganisms can lead to higher rate of reaction. So you could choose either one and note it down since they only ask for one other advantage. Part D. Figure 2.3 is a graph showing how the activity of alpha amylase extracted from barley seeds changes as the temperature increases from 10 degrees Celsius to 66 degrees Celsius. Part 1. Explain the effect of temperature on the activity of alpha amylase extracted from barley seeds as shown in figure 2.3. The keyword over here is explain, not describe. Hence, you have to talk about the details of this given graph. If you look at the graph, that is the graph they have drawn, not the graph I have drawn. The graph I have drawn is in red. Do ignore that for the time being and look at the graph drawn in black. If you look at the graph, the temperature increases throughout, and as the temperature increases till the optimum temperature is reached, the percentage enzyme activity also increases. Hence, you could mention that as the temperature increases, the kinetic energy increases because kinetic energy is linked to temperature, and once the kinetic energy increases, the proportion of successful collisions increases, or the frequency of successful collisions between the enzyme and the substrate increases till the optimum temperature is reached. That is why if the frequency of collisions increases, the enzyme activity will also increase because the enzyme is able to carry out its rate of reaction faster. And at the optimum temperature is where maximum enzyme activity takes place, because optimum temperature means the temperature at which the enzyme works best. After the optimum temperature, the enzyme activity decreases rapidly, and you could mention that the enzyme becomes denatured due to change in the shape of active site, leading to the substrate being unable to bind with the enzyme. If you look at the answers I have given over here, you could talk about increase in temperature leads to increase in kinetic energy. Higher the kinetic energy, more the number of successful collisions, hence increase in activity, until optimum temperature is reached at around 48 degrees Celsius. At temperatures higher than optimum, it leads to enzyme denaturation as active site changes shape. Therefore, the enzyme activity decreases. And that is how you explain the effect of temperature on the activity of alpha amylase. Hope it is clear. Part 2. Sketch on figure 2.3 the curve that would be obtained using alpha amylase enzyme that is heat stable. You could draw something like the above that I have drawn. The peak at 100% should be to the right of the original peak, and the line after the optimum temperature should be to the right and reach the horizontal axis. As I have drawn over here, it starts initially from the same point. However, the curve shifts towards the right, because if it is heat stable, that means the optimum temperature will lie towards the right at higher temperatures. And I have drawn something which closely represents the given question. My optimum temperature is towards the right, and the line drawn after the optimum temperature is also shifted towards the right. Basically, the whole curve is shifted towards the right if it is heat stable. And you could draw something like this. Question 3. T helper lymphocytes and Leydig cells are two types of mammalian cells. The main role of T helper lymphocytes and Leydig cells is to synthesize and secrete cell signaling molecules. T helper lymphocytes synthesize proteins known as cytokines. Leydig cells synthesize the steroid lipid hormone testosterone from cholesterol. Leydig cells also synthesize cholesterol. Part A. State one way in which cytokines are involved in an immune response. What you should remember is that cytokines stimulate the B cell production. Hence, if the B cell production is stimulated, there will be an increase in antibody production. It could also stimulate the macrophages to attack, and macrophages can carry out their role, which is the phagocytosis. Cytokines could also stimulate the response of killer T cells. Since the question only asks one way in which cytokines are involved, you could mention the best possible answer being it stimulates the B cell production, hence increase in antibody production. Or even you could mention the other key points. Part B. Figure 3.1 shows part of a mammalian cell. State with reasons whether figure 3.1 shows part of a Leydig cell or part of a T helper lymphocyte. If you look carefully at the diagram given, it shows the presence of many smooth endoplasmic reticulum. It does not have a rough appearance as no dark granules attached to the endoplasmic reticulum, hence it cannot be rough endoplasmic reticulum. Since there is the presence of many smooth endoplasmic reticulum, it should be a part of a Leydig cell. Why is that? Because the question, if you recall correctly, the question mentions that Leydig cell synthesizes lipids and lipid production is concerned with smooth endoplasmic reticulum. Therefore, the answer is it shows part of a Leydig cell, not T helper lymphocyte, because many quantities of smooth endoplasmic reticulum can be seen. And what is the function of smooth endoplasmic reticulum? It is concerned with steroid and lipid synthesis. If you recall chapter 1 of your biology textbooks, the function of smooth endoplasmic reticulum is concerned with lipid production or steroid production. And since the question mentions that Leydig cell synthesizes lipids, the answer is this diagram shows part of a Leydig cell. Why not T helper lymphocyte? Because T helper lymphocyte produces proteins which involves ribosomes. However, the diagram doesn't seem to show any ribosomes. Therefore, the answer is Leydig cell, and the reasoning is many quantities of smooth endoplasmic reticulum can be seen. That will give you 2 marks. Part 2. Underline the correct name for the type of image shown in figure 3.1 and explain your choice. If you remember the given image, it is not of a photomicrograph. Why is that? Because photomicrograph are those only taken through a light microscope. However, in this given diagram over here, you can see the intricate details of the cell. You can see the smooth endoplasmic reticulum as well as presence of mitochondria. Hence, it cannot be a light microscope that is used to view it, but an electron microscope. Hence, it is not a photomicrograph, because photomicrograph are those which we only call for those taken through a light microscope. Considering between scanning electron micrograph and transmission electron micrograph, the best possible answer is transmission electron micrograph. Why is that? Because you can see the structures within the cell or the ultrastructure of the cell. Example, membranes of mitochondria, endoplasmic reticulum, or even membranes. You cannot visualize the surface structure shown, hence it cannot be scanning electron micrograph. Scanning electron micrograph is only concerned with surface structures. However, this given diagram shows the intricate details or the other organelles visible inside the cell. Therefore, it should be transmission. Since this question is for 2 marks, 1 mark is for underlining and the other mark is for explanation. The explanation is you can see the structures within the cell or ultrastructure of the cell. Part C. Testosterone molecules and cytokine molecules are transported in the circulatory system to reach their target cells. Testosterone molecules are able to enter their target cells and bind to receptors within the cytoplasm. Outline one way in which testosterone molecules could enter their target cells. Key parts of this question are target cells and receptors present within the cytoplasm. Since there is a mention of target cells and cell surface receptors, the possible way would be endocytosis. So 1 mark is for the name of the process and the other mark is for what the process is about. So simply writing endocytosis would just land you 1 mark. The other mark is for pinching in of membrane to form a vesicle containing the testosterone molecule. Or you could mention it is an active process, hence ATP is required. Either of the two will give you the 2nd mark, because endocytosis is the pinching in of a membrane to form a vesicle containing the substance, and endocytosis is an active process. Therefore, ATP is required for any active processes. Part 2. Cytokine molecules are not able to enter their target cells. Suggest and explain why cytokine molecules are not able to cross the cell surface membrane to enter their target cells. Crossing the cell surface membrane, if you recall correctly, it involves crossing the hydrophobic regions, because a cell surface membrane is a phospholipid bilayer, and in a phospholipid bilayer there are hydrophobic tails. Since the presence of hydrophobic tails are there, that means there are hydrophobic regions. Therefore, one possible suggestion is that cytokines could be hydrophilic and water soluble, hence not allowed to cross the hydrophobic regions of the membrane. Another possible suggestion is that cytokines could be a large molecule, because large polar molecules are not allowed to pass through at all. You could also mention that there are no specific transport proteins to allow cytokines to cross. Since there is a mention of cytokine molecules not being able to enter the cell at all, there can't be transport proteins also present, because if there were transport proteins, then the cytokine molecules could at least take that pathway. Since the question refers to suggest and explain, so one possible suggestion is that the cytokines are hydrophilic and water soluble, and the explanation would be crossing the cell surface membrane involves crossing the hydrophobic regions. Since cytokines are hydrophilic and water soluble, it will not be allowed to pass through. That will land you 2 marks. The other possible suggestion and explanation is that cytokines are a large molecule and large polar molecules are not allowed to pass through the cell surface membrane. That will also land you 2 marks. Question 4a. Using a light microscope at a magnification of 400, it is possible to identify different types of blood cell in prepared slides of mammalian blood. Figure 4.1 is a key to identify different types of blood cell in prepared slides of mammalian blood. In figure 4.1, letters C, D, E and F represent 4 different types of blood cell. Identify the cell types C, D, E and F in figure 4.1. So they have given the keys and you just simply have to recognize what the cell type is. Since the question already mentions there are 4 different types of blood cells, you have to consider red blood cells and white blood cells. Red blood cells cannot be further divided into any sub-cells, whereas white blood cells, they form part of the lymphocytes, the monocytes as well as the neutrophils. And nucleus present are only in the white blood cells, because red blood cells, if you recall correctly, do not have a nucleus in order to maximize their space for oxygen transport. Therefore, nucleus present are white blood cells, nucleus absent are red blood cells. Hence C is a red blood cell. Let's look at cells D, E, F. If you look at 2A, large rounded spherical nucleus. Only lymphocytes have a spherical or a large rounded nucleus, hence D is a lymphocyte. Nucleus not rounded. Well, nucleus that are not rounded are either of monocytes or neutrophils. And you should remember that if the nucleus is kidney shaped, then it is a monocyte. If it is lobed, then it is a neutrophil. Again, kidney shaped nucleus is of a monocyte or a macrophage. Lobed nucleus belongs to neutrophils. Hence, cell D is a lymphocyte, cell E is a monocyte, cell F is a neutrophil. And all these three cells form part of the white blood cells. As you can see over here, I have identified the cell types: C red blood cell, D lymphocyte, E either monocyte or macrophage, F neutrophil. For red blood cell, you could also mention erythrocyte. Erythrocyte is also the same as red blood cell. Since the question refers to why a person who has infectious diseases may have a different number of white blood cells, you should state whether the white blood cell count would be higher or lower. So, in a person with an infectious disease, obviously there would be more white blood cells because more white blood cells are produced to attack the pathogen or the pathogen that causes the infectious disease. And since you have to explain, you have to talk about the result of immune response, because if white blood cells come in contact with a pathogen, immune response would occur, and immune response is where clonal expansion of either B or T cells occur. If there is an increase in B cells, it could lead to an increase in antibody concentrations, or you could also mention that there could be a possibility of an increase in macrophages. So those are possible points why blood taken from a person with an infectious disease may have different number of white blood cells. As part of a research project, a team of doctors and scientists carry out tests to investigate the effect of altitude on the ability of blood to carry oxygen. They assess the changes in the oxygen carrying ability of their own blood as they walk from 1,530 meters above sea level to 5,700 meters above sea level over a period of 14 days. 5,700 meters above sea level is an extremely high altitude. Table 4.1 is a summary of some of the results. The table given over here shows the percentage saturation of hemoglobin with oxygen in blood leaving the lungs, and also the volume of oxygen delivered to tissues by 100 centimeter cube of blood. And they have mentioned the day 1 at 1,530 meters and day 14 at 5,700 meters. Part B. Compare the results for day 1 with the results for day 14 in table 4.1 and explain the results shown for day 14 only. If you look at the words in bold, you have to compare as well as explain, and you have to explain the results only for day 14, not for day 1. So the comparison is there is a decreased percentage of oxygen saturation from day 1 to day 14 and a similar volume. It's almost a similar volume of oxygen delivered to tissues by 100 cm cube of blood. Those two are very important in the comparison part. And the explanation would be at high altitude presence of lower partial pressures of oxygen in the atmosphere. That you obviously have to know, because higher the altitude, lower the partial pressure of oxygen present. Therefore, in the lungs, less oxygen would bind to hemoglobin. This would lead to increased production of hemoglobin to compensate for reduced oxygen partial pressures, as the body needs more red blood cells or more hemoglobin to compensate for the reduced oxygen concentrations present at high altitudes. This then furthermore leads to an increased number of red blood cells, because higher the number of red blood cells, higher the hemoglobin content, because hemoglobin is present in the red blood cells, and higher the hemoglobin concentrations, that will lead to more oxygen delivered to the tissues in the body. As you can see, more hemoglobin available to bind with oxygen, hence similar quantity of oxygen delivered to tissues as at sea level. That is the possible explanation for results shown for day 14 only, because for day 14 it is 19.2 volume of oxygen delivered and day 1 is 19.3. So they are almost a similar volume of oxygen delivered to tissues by 100 cm cube of blood. And why is that? You have to explain it only for day 14. So you should talk about increased concentrations of hemoglobin to compensate for reduced partial pressures of oxygen. This then furthermore leads to an increased number of red blood cells. More the red blood cells present, more the hemoglobin available to bind with oxygen, hence similar quantity of oxygen delivered to tissues as at the sea level. That will give you 4 marks. Part C. The results of the research project have led to an improved understanding of diseases such as sickle cell anemia. Explain why sickle cell anemia is an example of a disease. Over here, you should mention the definition of a disease. A disease is nothing but a condition that affects the well-being or good health of a person, and it is characterized by a set of signs and symptoms. That will land you 2 marks straight away. You could also mention the impaired function, such as less oxygen taken up by red blood cells if a person has sickle cell anemia. Other key points are it has an adverse effect or lack of physical well-being. In order to get 2 marks, you simply have to state 2 key points, that is, a disease is a condition that affects the well-being of a person, as well as it is characterized by a set of signs and symptoms. That is enough to give you 2 marks straight away. Question 5. Each meristem cell in a leaf bud is able to grow and divide by mitosis to produce 2 daughter cells that are genetically identical to each other and to the original dividing cell. Part A. Figure 5.1 lists the stages in the mitotic cell cycle of a meristem cell. So the mitotic cell cycle, as you can see over here, it consists of the G1 phase, followed by S phase, followed by G2 phase, and then the stages of mitosis, that is, prophase, metaphase, anaphase, and telophase. Easier way to remember is that PMAT, P for prophase, M for metaphase, A for anaphase, T for telophase. And then lastly is the stage known as cytokinesis, which will leave you with 2 daughter cells. First part. Outline and explain the events occurring during S phase, metaphase, and anaphase of the mitotic cell cycle that are important in the production of genetically identical daughter cells. In S phase is where DNA replication occurs. And DNA replication, it ensures 2 genetically identical DNA molecules are produced, 1 DNA molecule for each daughter cell during mitosis. Since you have to outline and explain the events, the outline is that in S phase of interphase, DNA replication occurs. The explanation for why it is important in the production of genetically identical daughter cells is it ensures 2 genetically identical DNA molecules are produced, 1 DNA molecule for each daughter cell during mitosis. Then if you look at metaphase is where chromosomes line up at the equator of the cell. Explanation for that would be chromosomes are oriented in a manner that when they are split, sister chromatids will be shared out to the daughter cells equally. Because if they line up at the equator of the cell, they are equal distances away from the poles of the cell. Therefore, when they are divided, they would be equally split or equally shared between the 2 poles. And that ensures that the sister chromatids will be shared out to the daughter cells equally. And then lastly, anaphase. Anaphase is a stage of mitosis where the centromeres split and the separation of chromatids takes place. The explanation for why it would produce genetically identical daughter cells is the daughter chromosomes are then able to move to the opposite poles of the cell, thereby resulting in genetically identical daughter cells. Because when they split, each cell would receive equal numbers of the daughter chromosomes, and that results in genetically identical daughter cells. Part 2. Immediately after cytokinesis, daughter cells are not identical, even though they are genetically identical. Suggest a reason why daughter cells are not identical immediately after cytokinesis. A possible suggestion is that although the DNA molecules are equally shared among the 2, other organelles may not be shared equally. You could also talk about the unequal distribution of cytoplasm between the 2 cells. It is only the DNA molecules that are equally shared. What about the other organelles? So that is a key suggestion. Other organelles may not be shared equally, hence they are not identical immediately after cytokinesis. That will leave you with 1 mark. Part B. Some of the cells resulting from mitotic division in the young leaf from elongated cells that develop into xylem vessel elements. The xylem vessel elements are joined end to end to form xylem vessels. Suggest the structural changes that occur when elongated cells develop into xylem vessel elements and explain how these changes help xylem vessels to perform their function in transport. First, what is the function of xylem? The function of xylem is it transports water and mineral ions. And how does xylem do this? Well, there should be hollow empty cells with no obstruction to its flow. Therefore, the end walls of elongated cells are broken down. Since they are asking to suggest the structural changes, elongated cells would have end walls or sieve plates, and these would be broken down. to ensure the xylem vessel elements, they are hollow and no obstructions are present. Once the end walls of elongated cells are broken down, it would lead to uninterrupted continuous flow of water and mineral ions. Another structural change is that it forms lignified cell walls because only xylem vessel elements, they have lignin in their cell walls. And what does lignified cell walls do? It strengthens the vessel. So that is another structural change. Another structural change is the cells become hollow. And how do you explain this change? Well, if the cells become hollow, then it would lead to greater volume of water flowing in the vessels because no obstruction is present to its flow. Therefore, the points given over here would land you 4 marks straight away. Part 2. Figure 5.2 is a plan diagram of a transverse section through a dicotyledonous leaf. Add a label line and the letter X to figure 5.2 to identify the location of xylem tissue. This is a transverse section through a leaf, and in the transverse section of a leaf, the upper part of the vascular bundle, which is shown over here, is the xylem. Therefore, you should add a label line towards the upper region of this vascular bundle given, and that you should label with letter X to show the location of xylem tissue. And the lower part is the phloem. Question 6. In March 2019, a tropical cyclone in the southwest Indian Ocean caused widespread flooding in a number of countries. The flooding and the damage caused by the cyclone meant that many people were at serious risk of cholera. After the natural disaster occurred, many different areas reported outbreaks of cholera. Within a short time, the disease had spread widely and large numbers of people were affected. Part A. Cholera is caused by a bacterial pathogen. Name the bacterial pathogen that causes cholera. The bacterial pathogen that causes cholera is known as Vibrio cholerae. And a key thing you should know is that when you're writing the species that causes a certain disease, the starting letter of the species is always a capital letter. The second part, that is cholerae, should be starting with a simple letter. That is how you write the name of a species. Vibrio cholerae is the bacterial pathogen. Part B. Suggest and explain why the people affected by the cyclone were at serious risk of cholera. Over here, you simply have to give suggestions. There are no right or wrong answers, but your own thoughts. So, possible suggestions are either cholera was already present in the region, therefore leading to an outbreak. You could also mention contaminated water present containing the pathogen, therefore pathogen transmitted by water through contact with feces, no safe disposal of sewage and contaminated water supplied to people, unable to practice good hygiene as washing in contaminated water. So suggestions and explanations are these. Hence these points would land you with 3 marks. Part C. In addition to the standard treatment for cholera, antibiotics are recommended for people who are moderately ill or seriously ill with the disease. Doxycycline is one of the main antibiotics used for the treatment of cholera. Doxycycline enters the pathogen and binds to one of the subunits of the bacterial ribosome. This prevents growth and reproduction of the bacterial cell. Suggest and explain how binding of doxycycline to ribosome stops growth of the bacterial cell. Over here, ribosomes are mainly involved in the process of translation. If you recall protein synthesis, ribosomes are mainly involved in the process of translation. Hence, if doxycycline binds to the subunit of ribosome, it could lead to mRNA being unable to bind with the ribosome during the process of translation. Since it is also not mentioned which subunit is affected, you could mention the tRNA being unable to bind, because mRNA binds to a different subunit than the tRNA, as a ribosome consists of 2 subunits. tRNA binds to one part of a subunit and the mRNA binds to another part of the subunit. You could also mention, so the suggestions are that either mRNA is unable to bind or tRNA is unable to bind. Explanation would be codon-anticodon interaction is prevented. The overall process of translation is halted or prevented. Since the ribosome is concerned with protein production, proteins are unable to be produced or polypeptide synthesis doesn't occur. And that is how you explain why doxycycline leads to ribosome stopping the growth of the bacterial cell. Part 2. Penicillin, which used to be prescribed for the treatment of cholera, has a different mechanism of action to doxycycline. State which part of the bacterial cell is affected by the action of penicillin. Maybe it is there in your textbooks, and if you recall correctly, penicillin, it is concerned with preventing the formation of the cell walls of newly growing bacterial cells, because the penicillin's function is to prevent crosslinks forming in the cell wall of bacterial cells. Therefore, since the question refers to which part of the bacterial cell is affected, the best possible answer is cell wall, and that would give you 1 mark. Part D. Mozambique was one of the countries badly affected by the cyclone. As part of the effort to prevent a greater number of cases of cholera from occurring, 2 different approaches were taken. The first approach, approximately 900,000 doses of the oral cholera vaccine were sent to Mozambique, and a large-scale vaccination program was organized. Approach 2. Medical centers were set up in Mozambique to treat people with cholera. Suggest and explain how the 2 different approaches help to prevent a greater number of cases of cholera from occurring. This is another suggest and explain question, and it refers to the 2 different approaches used to prevent a greater number of cases occurring. Hence, possible suggestions and explanation would be: the vaccines provides active immunity to the people, and if the people are infected with the pathogen, people do not fall ill. If large-scale vaccination program occurs, that is approach 1, it would lead to herd immunity. And what is herd immunity? Herd immunity is where large number of people are vaccinated, leading to less spread among people, even to those unvaccinated too. Then if you look at approach 2, medical centers being set up. Medical centers set up aids in oral rehydration therapy to those already infected. If a person is infected with cholera and is not vaccinated, then medical centers could help that infected person by aiding in the oral rehydration therapy. And more are able to practice good hygiene. Medical centers can monitor the usage of antibiotics and provide effective medication. You could also mention that medical centers leads to less spread as more people are aware about the disease, cholera, and educated about its effects. And that will leave you with 4 marks. That is the end of this paper. Hope you did find it useful. If you did, why don't you leave a like and subscribe to keep this bio squad growing? Thank you and stay tuned for more.